I'm Dr. T, and welcome to my desktop. So continuing in our chain of videos on metabolism, let's jump into the wonderful world of the citric acid cycle, also known as the TCA cycle, aka tricarboxylic acid cycle, or Krebs cycle, depending on how you want to refer to it. It's all the same thing. So here's a nice, I'll be a little bit busy diagram of this guy. So what's going on over here? If you remember from glycolysis, you ended in pyruvate. Now that pyruvate could get sidetracked over to fermentation, which we talked about in the last video. However, that loses a lot of energy. If you really want to get the energy out, you're going to get a ton more energy if you can react this further. And so you got a couple things you can do. You got this kind of little side jump over here that we're not really going to deal with. That's if you want to build up more citric acid cycles, because you kind of want to go around that. But most of the pyruvate is going to get turned into acetyl-CoA. Now, acetyl-CoA is a rather large cofactor that is grabbing a hold of what is basically two carbons. So you're going to take the pyruvate, you're going to lose one carbon, you're going to get a CO2. Whenever you get a CO2 off, that's a one-way reaction. It is very, 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 very hard to go backwards to the point where basically you can't. So when you did fermentation, that was one of the reasons why humans, we do lactic acid fermentation. It's much easier to go around. You can do gluconeogenesis. When the yeast give off that CO2, it's done. You can go feed ethanol and turn it into acetyl-CoA, but you can't turn it back into glucose. Side note, that CO2 given off during the alcoholic fermentation is also why alcoholic beverages are fizzy. Just side note. But jumping back into citric acid cycle. So we're going to give off the CO2. Of course, that's going to be exhaled. Interestingly enough, the CO2, the oxygens from that, do not come from the oxygens we breathe. At least not directly. The things get shuffled around, don't get me wrong. But those are coming off of what's being oxidized. Okay, so we're going to go pyruvate to acetyl-CoA. Now that acetyl-CoA could have a couple other sources, or alternatively, it could have a couple other fates. We'll talk about fatty acids. That's going to go to and from acetyl-CoA. So they are metabolized into acetyl-CoA, or they're going to be built from acetyl-CoA. And some of your amino acids go into acetyl-CoA. Anyways, we're going to take acetyl-CoA. And usually this guy is, once again, put as kind of its own thing because it's really a hub of a lot of chemistry. Now, that acetyl-CoA is going to get reacted with an oxaloacetate, forming a citrate, giving the name citric acid cycle. And this is going to go through a series of different reactions. We're going to give off some carbon dioxide in the process. We're going to give off two. Essentially, we're going to take that pyruvate. We're going to have three carbons. We're going to give off one going to acetyl-CoA, and citric acid cycle, we're going to get rid of the other two. We're going to use this up. Now, it turns out those aren't actually the exact same two carbons. Some of them are coming from that initial oxaloacetate, but the net result is the same. Carbons are carbons. Okay, so we're going to go through this. We're going to produce a fair amount of NADH. Notice how we've got multiple there. We actually happen to have a GDP taking place instead of an ATP that's getting turned into a GTP instead of an ATP. So it's actually an interesting one. This QH2, this is part of electron transport chain. We'll talk about in a little bit. Basically, this is more electrons. So you're getting these high energy electrons. This is slightly less high energy electron, but still high energy. Oh, we're bigger. Yeah. Okay. So the general idea, though, at this point is that we're getting CO2 away, and we're getting a lot of high energy electrons. Okay, so that's going good. Now, what are we going to do with those? We're going to take that over here to the electron transport chain. We actually have two electron transport chains. Well, we don't. Plants have their own electron transport chain, which I'll talk about in a different video. But here's kind of the gist for ours. So citric acid cycle is taking place inside the mitochondrial matrix. So remember, your mitochondria has kind of two layers. It actually has more than two, but two major layers. You have the outer membrane, and then you have this inner membrane. The gap between them is called the inner membrane space because we're not original. And the inner part of the inner membrane is the matrix. And citric acid cycle is taking place in the matrix. Glycolysis would have taken place in the cytoplasm, completely outside the mitochondria. The pyruvate would have then been shipped in. Pyruvate and acetyl-CoA. So anyway, so what we're going to do is we have a series of complexes. We call this complex 1. There's a 2, which is hanging out over here. This complex 2, by the way, the succinate going to fumarate, that is actually this guy right here. So that's this step in the pathway. Okay, so we're going to have the high energy electrons from the NADH. They're going to go into complex 1. You're going to have more energy going into complex 2. And it's going to go through, 1, 2, 3, 4. The ones going into 1 don't go into 2. So we're going to go from 1 to 3 to 4. Now, okay, what's the big deal? So after 4, we're going to take some oxygen. This is why you need oxygen. You're going to take oxygen, you're going to dump the electrons onto it. It's going to pick up some hydrogens, some H pluses, and going to make a water molecule. In the process, each time we're doing this, we're recycling our NADH, making back NAD, so we can go back and do the cycle. Okay, that's nice. Why do I care? Well, during this pumping phase, as electrons are transferring further, we're moving hydrogen plus from the matrix to the inner membrane space. We're actually doing something that if you would have heard any of my lectures from Survey 1, I would have said cannot happen. You can't move just one ion by itself. Well, actually, you can. So your cells are moving the cation. They are not moving the anion. The anion is staying right there. This then results in that you actually get a charge buildup between both sides of the matrix. You're basically building a capacitor. Not to mention you've got concentration gradient. You're setting up a bunch of energy. So you're taking the energy from these high energy electrons that had previously come from your reduced carbohydrate or reduced whatever your sugars were, or your fatty acid, whatever your source material was that went through metabolism. You then take that energy, put it into high energy electrons. Those high energy electrons then go into water. In the process, they shut off their energy in the form of this potential between these two sides of the membrane space. Now, complex 5, someplace called that, others call it ATP synthase. You'll hear it called FATPase, just depends on where exactly you're looking at. Allows the protons to flow backwards. So it's basically short circuit, well, it's not short circuiting, but it's allowing the cycle to continue and to go back. And as it does, this literally rotates. And it's going to take ADP plus some phosphate and make ATP. So this is going to allow us to take the energy from these high energy electrons and turn them into a far more usable form, the ATP. Now, yay, this allows us to do what we need to do. The ATP can then get shipped throughout the cell as need be. We can also use this gradient in other sources, so it's not just mitochondria that use this. This kind of gradient fashion will be used for Flagelman bacteria as well as other sources. Even eukaryotic cells will use the gradient directly. This can be used to move stuff if need be. So you don't have to go to ATP. You can short circuit this a bit. And that's actually an interesting side thing. There's things such as brown fat or brown adipose tissue. Has lots of mitochondria, that's why it gives it a brown color. And they actually have short circuits in here, what we call decouplers. And that will allow the hydrogen cations to flow back. And just like you have a short circuit in wiring, when you have a short, it creates heat. So brown fat or brown adipose tissue is found in hibernating mammals. It's found in human infants. And it's just there to take fuel and make heat so that you can keep warm. As adults, we don't have it. Okay, and yeah, that's the best pictures we got. Long article. Okay, so that's electron transport chain. Next time, we'll look more into a few of the other kind of side pathways. See you next time.